Okay, so welcome to this next video in which we are discussing lipoproteins and the transportation of lipid molecules around the bloodstream. Okay, so we are now in the process of discussing the digestion and absorption of fat so that we can see the formation of the first type of lipoprotein in our physiological pathway, which is the chylomicron. Okay, right. So we have had the bile arrive from the liver, and we've had the enzymes arrive from the pancreas. Okay, so the bile contains a huge amount of lecithin, also known as phosphatidylcholine, which helps in the emulsification of the fat, which means breaking great fat globules down into loads of little fat droplets. Okay, now we want to see the enzymes which are going to digest the different lipid molecules within these little fat droplets. Okay, so the first one we've seen is pancreatic lipase, which acts on triacylglycerol molecules, okay, and cuts two long-chain carboxylic acid molecules off these triacylglycerol molecules, converting them firstly into a diacylglycerol molecule and then finally into a 2-monoacylglycerol, where the remaining acyl group is attached to the alcohol group that comes off the second carbon of the glycerol molecule. Okay, now let's also discuss the digestion of the phosphatidylcholine molecules which you have within the lumen of the intestine. Okay, and more generally, this is going to be how phosphoglycerolipids are digested. So remember, phosphatidylcholine was an example of a phosphoglycerolipid. So you have this glycerol molecule with two long-chain carboxylic acids esterified to the first and second alcohol groups of the glycerol molecule, and then a phosphate group linked via a phosphoester linked with the glycerol molecule's third alcohol group, and also off that phosphate group you then have some head group such as choline. So let's colour this in. Here's the phosphate group in purple. Here's the additional head group in blue, that could be choline for instance. Here is the glycerol molecule in green. And here are our long-chain carboxylic acids in orange here. Okay, so this is our phosphoglycerolipid molecule, our P for phospho, G for glycero, and L for lipid molecule, our PGL. Okay, so basically, these are also going to be digested in the lumen of the intestine, and the enzyme that digests the phosphoglycerolipids is known as phospholipase A2. Okay, so PLA2 is short for phospholipase A2. So the P is phospho, the L is lipase, and then the A2 is just A2. And basically, these enzymes are secreted by the pancreas into the duodenum, and what they're going to do to these phosphoglycerolipids is they're going to cut the ester link between the long-chain carboxylic acid and the second alcohol group of the glycerol molecule. So they're going to cut this bond here, and therefore they're going to produce two products from performing this reaction. Okay, so you'll have the bigger product, which is what's left of the phosphoglycerolipid after you have removed the long-chain carboxylic acid that is in position 2. So here we have a long-chain carboxylic acid still in position 1, then we have our glycerol molecule, and then we have a phosphate group coming off the alcohol group of the third carbon, and then finally we have this head group off the phosphate group up here. Okay, now basically, this molecule has a different name depending on which phosphoglycerolipid you were talking about. So, for instance, if this head was choline, what you would call this is a lysophosfatidylcholine molecule. However, if it's a different group that you've got here, it would instead be called lyso and then whatever that phosphoglycerolipid was. So I'm just going to call this a lysophosphoglycerolipid, although that isn't really standard nomenclature. Okay, so basically all we've done is cut the second long-chain carboxylic acid off the glycerol molecule. Okay, right. So if we're talking about phosphatidylcholine, which is our main example of a phosphoglycerolipid that you're going to be absorbing from the lumen of the intestine, okay, then this molecule here would then be called lysophosphatidylcholine. Okay, right, so once you've chopped off that single long-chain carboxylic acid, it's called lysophosphatidylcholine. And then, of course, the other product of this will be that you get this long-chain carboxylic acid, which we'll just call a free fatty acid. Okay, right, so this is the digestion then of these phosphoglycerolipids. Okay, the final thing to talk about is the digestion of cholesterol esters, okay? So let me draw one of these cholesterol esters. Okay, so remember, cholesterol has this steroid structure within it, so it's got these 4 carbon rings. Okay, so here's the third 6-membered carbon ring, and then you've got your 5-membered carbon ring here. Okay, and then in cholesterol you have a double bond here, a methyl group there, a methyl group there, and then this 7-membered group coming off up here, like so. Okay, and then you would have an alcohol group here in the pure cholesterol, but in a cholesterol ester, what you have is a long-chain carboxylic acid, so a fatty acid, esterified to that alcohol group of the cholesterol molecule. So this is what's known as a cholesterol ester. Okay, and basically these cholesterol esters are going to be split down into cholesterol, okay, and then the long-chain carboxylic acid, so plus a free fatty acid, okay, and this hydrolysis reaction is going to be performed by an enzyme known as cholesterol ester hydrolase. Okay, so this enzyme will be secreted by the pancreas into the duodenum, and it will then break down cholesterol esters into cholesterol and free fatty acids. Okay, so that's cholesterol ester hydrolase. Okay, right. So we've now broken down the major lipid components that are within the diet. We've got the cholesterol esters, we've got the phosphatidylcholine, and then we've also got the triglycerides being broken down into these digestion products. And now what's going to happen is these digestion products need to be absorbed across the epithelium of the small intestine. So basically, there is a problem for digestion, another problem that they faced, which is that all of these digestion reactions that we've just talked about, for instance, the breaking of the ester links within a triacyl glycerol to yield a 2-monoacyl glycerol and 2 free fatty acids, the breaking down of the phosphatidylcholine molecules to yield lysophosphatidylcholine and free fatty acid molecules, and the breaking down of cholesterol esters to yield cholesterol and free fatty acid molecule. All of these reactions are actually reversible, so the enzymes can catalyze them both ways. So basically, if you don't want your products of digestion to be reconverted back into the original things, then you need to remove them from the lumen of the intestine, from the vicinity of these enzymes, pretty quickly. Okay, now this is the function of the bile acids. So I told you we were going to come back to the bile acids, which are the other major component of bile other than the phosphatidylcholine. Okay, so let's now talk about bile acids in a bit more detail. So basically, most bile acids are based on the structure of cholic acid. Okay, and cholic acid itself is a derivative of cholesterol. So previously I told you that the bile acids were all derivatives of cholesterol, which is true, and most of them have their structure based on this molecule cholic acid, okay? So, let me now show you the structure of cholic acid, and then you'll see how it's got a very similar structure to cholesterol. Okay, so again, it's a sterol, so it's got the steroid structure, so we'll draw that first. So here are the 4 carbon rings that are linked together. Here's the second 6-membered ring, the third 6-membered ring, and then the 5-membered ring right at the end here. Okay, like so. And then in cholic acid, what you then have is the alcohol group coming off here to make it a sterol, but then in addition you have methyl groups here and here, just like in cholesterol. Then what you have, which cholesterol doesn't have, is two alcohol groups that are in addition to this alcohol group over here. So you have one here and one here. You don't have the double bond there, so that's not that present in cholic acid. And then what you're going to have is a 5-carbon carboxylic acid molecule on the side here. So 1, 2, 3, 4, 5, okay? And then this 5th carbon is going to be the carboxylic acid group here. So that is the structure of cholic acid. Now, cholic acid itself isn't usually secreted as a bile acid, but conjugates of it are. So what you do is you add things onto this carboxylic acid group to turn them into bile acids. So let me show you an example of what you can add onto cholic acid. So something which you can add on to cholic acid is something called taurine. Okay, now the proper name for taurine, or the organic chemist name for taurine, would be 2-aminoethane and then sulfonic acid. So 2-aminoethane, and then it should all be one word, but never mind, sulfonic acid. Okay, right, so basically the structure of taurine then has a 2-carbon structure. Okay, that's the ethane. Then we know we have an amino group of one of these carbons, okay, the second one, so we'll call this the second one here, and we'll put our amino group coming off there. And then off the first carbon we have to have this sulfonic acid group. Now what's the structure of a sulfonic acid group? Well, basically you have a sulfur atom with 2 oxygens doubly bound to that sulfur atom, and then an alcohol group here. Now you might ask, well, why is this called an acid? Well, basically, just like when we were looking at the phosphate group earlier, and I said that the structure was similar to that of a carboxylic acid group, if we look at this structure here, where we have a sulfur atom double bound to an oxygen and then with an alcohol group coming off it, that's actually got a very similar structure to a carboxylic acid group. And in fact, the proton of this alcohol group here can in fact detach off that alcohol group, and therefore can be donated away into the solution, making this an acid. Okay, so basically what you can do is you can join cholic acid with taurine to make an example of a bile acid. Okay, and this isn't the only exclusive bile acid, but this is a nice example of a bile acid. Okay, so we're going to now join them together. So let's start by drawing out our cholic acid structure again. Okay, so here's our steroid structure, once again with its 4 carbon rings here, 3 6-membered carbon rings and 1 5-membered carbon ring. Then this alcohol group here is unaffected. You might have thought that maybe we're going to esterify this sulfonic acid group onto one of the alcohol groups, like we've esterified carboxylic acid groups and phosphate groups onto alcohol groups. But in fact, what we're going to do is we're going to bind this carboxylic acid group of the cholic acid to this amino group of the taurine molecule via an amide link, basically. And that will leave the sulfonic acid group intact, basically. So here is an alcohol group up here, the methyl group, and now we've got this 5-membered carbon here, and then off this we will have the carbonyl group, and I'm now reverting to drawing a skeletal structure. So here's the carbon of the carboxylic acid group with the oxygen double bound to it, and now it's going to be bound to the nitrogen atom of the taurine, and it will have a hydrogen coming off it, and then it will have 2 carbons like so, and then finally we'll need to show the sulfonic acid group. So here's a sulfur doubly bound to an oxygen, and then another oxygen double bound to it, and then an alcohol group here. Okay, and the name for this example of a bile acid is taurocholic acid. So this is taurocholic acid. So what is the actual function of bile acids then? Well, basically, when you secrete them into the intestine through the bile duct, which enters into the main pancreatic duct, which then dumps its contents into the duodenum via the major duodenal papillae, Okay, so basically what happens is the bile acids all join together to make structures called mixed micelles. Okay, and these are basically little spheres of bile acids. So let me show you the structure of a mixed micelle. And after looking at lipoproteins and also those globules of fat, this is a structure that you can probably dream up of your own accord. Okay, so basically what we're going to have is a sphere which is made up of loads and loads of bile acid molecules. So basically, let's imagine that this rectangle here represents a bile acid molecule. What you're going to have is a monolayer of bile acid molecules going round and making the surface of this sphere like so. Okay, so you'll have a complete surface made out of these bile acids like so. Okay, and this is what a mixed micelle is basically. And this special group here, such as the sulfonic acid group, and although taurocolic acid isn't the only bile acid that you can have within bile, there are others. Basically, they all have some sort of group along the lines of this, which will generally lose its proton when it goes into the intestine, because the intestinal contents is generally quite alkaline. Okay, so generally it will lose its proton, gain a negative charge here, and therefore it will be negatively charged. It will be polar. Okay, and that group will face the external world basically. So this molecule will be oriented with this facing out towards the external world and this portion facing in towards the middle. Okay, and that will mean that this polar portion is interacting with the water molecules, and all of the hydrophobic portions can be facing in towards the center basically. Okay, so we'll colour these bile acid molecules in pink here. Okay, right, so this is at the moment a micelle. It's going to become a mixed micelle because basically what's going to happen is the products of digestion are going to join the micelle, and therefore you're going to get all sorts of other lipid molecules apart from the bile acids within these micelles. Okay, and this is how you're going to protect the products of digestion from being reacted upon by their enzymes to convert them back into the original reactants, which we want to stop, remember. Okay, so basically the products of digestion such as the free fatty acids, the cholesterol molecules, the lysophosphatidyl molecules, and also the 2-monoacylglycerol molecules, these are all going to join the mixed micelles. So let me try and draw some of these. So you're going to get your long-chain carboxylic acids joining in. You're going to get cholesterol molecules joining in here. You're going to get your monoacylglycerols joining in here, and they're all going to be added in to the mixed micelle. And basically now, these mixed micelles are going to deliver the products of digestion to the enterocytes. Okay, so to the cells which line the surface of the small intestine. So the products of digestion have mixed into the mixed micelle, and now this micelle is going to take its products of digestion to an enterocyte which is on the surface of the small intestinal wall. Okay, so let me show you the structure of an enterocyte. Okay, so here is an enterocyte. So this is an epithelial cell that sits on the surface of the small intestinal wall. And basically, their apical surface is not just flat. Instead, they have these little finger-like projections out from their apical surface. And these are what are known as microvilli. Okay, like so. So these are called microvilli. Now, what's going to happen is this is the luminal side, so this apical face faces the lumen of the small intestine. So what's going to happen is these mixed micelles, which I'll just draw as a little dot here, which I'll colour in pink, these are going to come and go down the gaps between the microvilli. So this little mixed micelle will come down here into this gap between neighbouring microvilli, and then what's going to happen is the products of digestion that the mixed micelle is carrying will go from the mixed micelle into this enterocyte. Okay, so this is a enterocyte. Okay, and it's just an epithelial cell, a simple columnar epithelial cell that is on the surface of the small intestinal wall. Okay, now, what happens within the enterocyte? Well, basically, in the enterocyte, the processes of digestion are going to be reversed. You are going to resynthesize triacylglycerols. You're going to resynthesize cholesterol esters. You're going to resynthesize phosphoglycerolipids from the lysophosphoglicerolipid and the free fatty acid molecules. Okay, so let's get another piece of paper and discuss this further. So, basically, in the enterocyte, you have an intracellular organelle called the smooth endoplasmic reticulum. So I'm just redrawing my enterocyte here. Okay, so the products of digestion, which remember are these free fatty acids, so I'll just remind you what the products of digestion are. We're getting free fatty acid molecules, we're getting cholesterol molecules, which I'll just denote as C. We are getting 2-monoacylglycerol, okay, and we're also getting the lysophosphoglicerolipids. Okay, so I think I should actually just draw little pictures. So the 2-monoacylglycerol is a glycerol molecule with a single acyl group still attached to it, and the lysophosphoglicerolipid is a phosphoglycerolipid with that middle acyl group removed from it. Okay, so here's the head, and I'm calling it a lysophosphoglicerolipid to keep it nice and general, although the main phosphoglycerolipid will of course be phosphatidylcholine. So let me add some colour onto here. So here in orange is the free fatty acid, here's an acyl group, here's another acyl group, then we've got 2 glycerol molecules, one there, and then we've got this head of the lysophosphoglicerolipid that's attached to the phosphate group there. Okay, so all of these products of digestion are coming into the enterocyte, and basically within the cytoplasm of the enterocyte you have an intracellular organelle, and the nucleus is down here, so that's not meant to represent the nucleus. And this intracellular organelle is called the smooth endoplasmic reticulum. Okay, and whilst the rough endoplasmic reticulum is very much so involved in the synthesis of proteins and the processing of proteins, and it's called a rough endoplasmic reticulum because its surface is very rough, because it's covered in ribosomes, basically. Okay, the smooth endoplasmic reticulum is far more involved in the production and the metabolism of fat molecules. Okay, so what's going to happen is in the smooth endoplasmic reticulum, you are going to get these free fatty acid molecules arriving from the mixed micelles that are delivering them to the enterocyte, and you're also going to get these 2-monoacylglycerol molecules arriving, and basically what will happen in the smooth endoplasmic reticulum is you will resynthesize those to triacylglycerols. Okay, so the entire purpose of the digestion was just to get them across this apical membrane of the enterocyte, basically. So, let me now colour in the picture of a triacylglycerol. So here are our 3 acyl groups that are bound to the glycerol molecule here in green. Okay, we're also going to get quite a bit of our cholesterol resynthesized into cholesterol esters. So I'll just draw a little structure for this. So here is our cholesterol steroid structure. So here are carbon rings. So there's our second 6-membered carbon ring, our third 6-membered carbon ring, and then our 5-membered carbon ring here. Then we have a double bond there, 2 methyl groups here and here. Then our 7-membered carbon side chain up here with a methyl group coming off it. And then down here we have this alcohol group, which is now going to have a long chain carboxylic acid linked to it. Okay, So we're going to take 3 fatty acid molecules plus cholesterol, and we're going to resynthesize them into cholesterol esters. Okay, now not all of the cholesterol will be resynthesized into cholesterol esters, because not all of the dietary cholesterol that you took in was originally cholesterol esters, so some of it will remain as free cholesterol. Okay, and then finally we're going to take the lysophosphoglycerolipid, attach another long-chain carboxylic acid here to reconvert it back into a phosphoglycerolipid, which is good news for me because it means I can now use standard terminology rather than non-standard terminology. Okay, so here is our phosphoglycerolipid here. So here is our phosphate group, here is our glycerol backbone for the phosphoglycerolipid, and here are 2 long-chain carboxylic acid groups which are attached to the first and the second alcohol groups of that glycerol molecule. So this is a phosphoglycerolipid. Okay, so these are all being formed in the smooth endoplasmic reticulum of these enterocytes. Now what's going to happen basically is that the enterocytes are going to package these products that they've now produced into structures called chylomicrons, which, remember, are massive great lipoproteins. So basically, you are going to produce from these molecules chylomicrons. Okay, so let's now revise the lipoprotein structure. Okay, and we'll talk about which specific apolipoproteins chylomicrons have within them. So chylomicrons are basically absolutely massive lipoproteins. So they're between 50 and 200 nanometers. They're the giants of the lipoprotein world. So they will have huge phospholipid monolayers, which will be mainly made up of phosphatidylcholine. Okay, so let's have this outer layer here representing the phospholipid monolayer, which, as I say, is mainly made up of phosphatidylcholine, also called lecithin. So this is the phospholipid monolayer, although there will be some other phosphoglycerolipids, and maybe even some sphingomyelins as well, the other forms of phospholipids that are not phosphoglycerolipids. Okay, so let's colour that in purple here. So I'll just give it a little bit of a shade in, like so. So this is the outer phospholipid monolayer. Right. Then, of course, embedded into the phospholipid monolayer, you have apolipoproteins. Now, because of the sheer size of these chylomicrons, you're going to have multiple apolipoproteins, basically. So I'll have some of them in turquoise, maybe, and some of them in blue. Now, remember, I listed out for you the different apolipoproteins that lipoproteins can have within them. Okay, so let me now tell you which of those apolipoproteins you actually find within chylomicrons. So basically, within chylomicrons, the main 3 apolipoproteins you find are apolipoprotein B48, then also apolipoprotein C2, and then finally apolipoprotein E as well. Those are the main 3 that you're going to find within chylomicrons, phospholipid monolayers. There are 2 others which you can find within the phospholipid monolayer of chylomicrons. You can also find apolipoprotein A4, okay, and then also apolipoprotein C3. Okay, so those are to a lesser extent. These are the main 3 that you find within the phospholipid monolayer of chylomicrons. Now, in the core then of the chylomicron, you will have a huge number of triacylglycerols. Remember, this is the main dietary intake of fat molecules that you're going to be getting. Triglycerides are the main form of dietary fat, so you've got a huge number of triacylglycerols within the lipid core. But you will also have cholesterol esters, which I'll just denote as CE. And in addition to that, you're also going to have a few cholesterol molecules within the chylomicron, chylomicrons, and they will be embedded within the phospholipid monolayer, because remember, they have alcohol groups which aren't involved in ester links, and those alcohol groups are slightly polar, so those can join with the phospholipids and have the polar groups sticking out towards the outer surface, just like the phospholipids have their polar group sticking out to the outside world. So, here then is the structure of a chylomicron. Now, basically, these things are not going to be passed straight into the blood from the enterocyte. Instead, the enterocytes are going to manufacture these things within their cytoplasm, okay, and then they're going to send them out, and they're going to go into lymphatic vessels, okay. So, let's imagine that our enterocyte is sitting on some villus of the small intestine. So, let me just show you a villus here. So basically, the mucosal layer of the small intestine is not flat, but instead has these little outward projections known as villi. Okay, so here are all the enterocytes sitting on top here. Okay, and I haven't shown the microvilli, but of course they will have microvilli like so. And basically, if we use, for example, this as our example enterocyte that we are studying, so let's say this one in purple is this star up here, okay, then basically down the center of the villus you will have a large not too large, but an arteriole running, and you'll also have a venule, okay. And basically you'll then have capillaries going between these two. And of course, you might think that the chylomicron will just go into one of those capillaries, then it will go into the venule, and it will be taken to the liver along with everything else through the hepatic portal vein. But instead, the chylomicrons take a different path. They go through the lymphatic vessel. So the third vessel that you have running within every single villus is a vessel known as the central lacteal. Okay, and it's effectively a massive great lymphatic vessel. So every single villus has a central lacteal. Okay, and basically the chylomicrons go from the enterocyte into the lymphatic vessels, and then they'll drain into this central lacteal. And then where does the central lacteal drain? Well, basically all of the intestinal lymph goes to a structure known as the cisterna chyli. Okay, and we'll discuss the cisterna chyli and where it will eventually drain into in the next video.